All right, hello everyone. My name is Leavitt. I work at the University of Utah for Dr. Baldomero Oliveira, where I work on this platform, Pharmaconomics. If you're interested in a more biology-centric talk, I welcome you to go to YouTube and search for Constellation Pharmacology. The system that we study with this technique is the dorsal root ganglion. This is considered the sensory organ of the body. It sits at regular intervals along your spinal cord, where it sends projections to the periphery where you sense pain, heat, cold, and touch. It then transmits this information back to the central nervous system. Right here, I'm showing you a cross-section of the dorsal root ganglion. Each green dot is an individual neuron, but getting access to this three-dimensional structure is very difficult. So we turn to a technique where we create a primary cell culture. We break up this three-dimensional structure very gently, keep the cells alive, and plate them. We can see that the live cell culture is diverse. We have everything from small, medium, and even large-sized neurons. In addition to this, we have genetic markers and structural markers which identify to us cells which sense pain. There's green pain and red pain. If you want to know more, watch my YouTube video. But in addition to this, we want to understand which ion channels are present on these neurons. Ion channels are essentially switches which turn the neurons on or off. For example, heat will turn on this ion channel, TRPV1. Cold will turn on the TRPM8 channel. And noxious sensation will open the TRPA1 channel. To understand which cells have these ion channels without killing them or fixing them, we turn to the technique Constellation Pharmacology, where we use pharmacology to answer the question of which neurons have these switches. For example, we have pharmacology which comes from wasabi, which activates TRPA1. We have pharmacology which comes from mint plants to activate TRPM8. And we have pharmacology from spicy chili peppers which activate TRPV1. So to understand which of these cells have these ion channels while still keeping them alive, we load the cells with the dye indicative of calcium. This is going to tell us when these ion channels open. So for example, when I apply AITC here, we can see the cells turn red, and we capture that information down here on the bottom left. Here, when I apply menthol, cell 1 is going to give us a response, but cell 2 and cell 3 do not. This really encapsulates how diverse and heterogeneous the cells are. Here, I'm only showing you examples from 3 neurons. We're collecting data from all 3,000, 4,000 cells simultaneously, and we are turning it into a time series, which you're seeing here on the bottom. Now what we do is we form a facile cell type classification, kind of a fake cell classification where we classify cells based off of what we think is important. So let's just take a break here and catch up. First, we use Constellation Pharmacology. It's a combination of fluorescent imaging, calcium imaging, and pharmacology. We collect a lot of data and we put it through Cell Profiler and our in-house software, ProcPharm. Here you can see researchers going through and labeling the data. They want to understand what responds and what doesn't respond. This creates an awesome labeled data set, which we can then automate these tasks using LSTMs and convolutional neural networks. This helps us automate our facile cell type classification that I briefly introduced you to. And now we turn to more difficult techniques like single-cell transcriptomics. Now we identify individual neurons from this cell type classification and pick them up with these pipettes. It's a technique I like to call farming because we use pharmacology to guide our transcriptomic efforts. Now we get very complex and interesting data set. We're getting about 17,000 genes per cell, and we can start to answer questions like, What are the definite cell type classifications? Not just the facile cell type classifications, not these fake cell type classifications. Can we identify what their definite role is? Such as, do they project to the skin? Do they project to the muscle? Now we can move into more difficult experiments such as molecular target identification. For example, can we identify molecular targets which are turned on or off during disease states? Here we're working on a chronic constriction injury model where we injure some of the dorsal root ganglion neurons while we don't injure others, and then we compare the two and pick up these cells for transcriptomics. We also are making great progress in drug target identification. So now you can take known and unknown compounds, put it onto our plate of onto our cells, and we can identify top candidate genes that these drugs are targeting. So now we can take unknown drugs or even known drugs, put it into our assay, and give you identification of the targets that your drug is hitting. We're doing this with a lot of really fun and interesting data mining operations, and I have slides prepared, but I'm going to stop here to take questions. I prepared slides for how we automate our cell type classification, and then I've also created some slides on molecular target identification. Thanks for listening to my talk. Feel free to reach out to me on LinkedIn if you want to know more. I'm currently in customer validation of my business model hypothesis, so if this is interesting at all to you, please reach out. Thank you.